Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 2 of your OpenSTAX book, How We See the Invisible World. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. The learning outcomes for this chapter include familiarizing yourself with the size of various specimens, being familiar with terms such as resolution, magnification, and refraction index, comparing and contrast, light and electron microscopes, comparing transmission to scanning electron microscopes, and the different types of stains. We begin today by examining the relative sizes of microorganisms. Microorganisms come in a broad spectrum of sizes. The typical virus measures about 100 nanometers, which is 10 times smaller than a typical bacteria. That typical bacteria, at approximately 1 micrometer, is at least 10 times smaller than a typical plant or animal cell. An object must measure about 100 micrometers to be visible without a microscope. The graphic attached to this slide will help you gain some perspective of size of a variety of different cell types. Properties of waves. Here we will pause and take a little side jaunt into the world of waves, not waves from the ocean, but in this case, waves of light. Light is the primary tool used by a microscope to illuminate a specimen, and it travels in waves. A wavelength is the distance between one peak of a wave and the next peak, while the amplitude is the height of each peak or depth of each trough. The frequency is the rate of vibration of the wave or the number of wavelengths within a specified time period. As light travels from whatever source produced it, it travels in this up and down wavelength pattern. As energy is increased to the light, the frequency increases and the wavelength decreases. As we dig deeper into the properties of light, we'll examine reflection, absorbance, and transmission. Reflection occurs as waves of light bounce off of a material, while absorbance is the opposite. The material captures the energy from a light wave. Transmission describes the process where a wave travels through a material. Refraction. The refractive index is a measure of the light bending ability of a medium. The light may bend in air so much that it can actually miss the small opening of a lens on a microscope. To compensate for this, microbiologists will often use a tool, immersion oil. Immersion oil is used to keep light from bending and missing the lens. Light travels in wavelengths that we refer to as the electromagnetic or visible light spectrum. Visible light is a narrow range of electromagnetic radiation that can be described using the acronym ROY-G-BIV, which stands for red, orange, yellow, green, blue, indigo, and violet, or the colors of the rainbow. The longer the wavelength, the less energy light has, and vice versa. In this picture, red light, which has the longest wavelength, is traveling slowest and has the least amount of energy. Magnification, resolution, and contrast. Magnification is the ability of a lens to enlarge the image of an object when compared to the real object, while resolution is the ability of the lenses to distinguish 2 points. A microscope with a resolving power of 0.4 nanometers can distinguish between 2 points as long as those 2 points are further than 0.4 nanometers away from one another. Shorter wavelengths of light provide greater resolution, as does increasing your numerical aperture on your microscope. The higher the aperture, the better the resolution. This is a picture of a compound light microscope, the same type that we use in our labs. Take a moment to review each labeled part. The oil immersion lens. The oil immersion lens is used to decrease refraction. Oil is used between the lens and the specimen. Oil has very nearly the same refractive index as glass, and therefore reduces the amount of light lost as light travels from the light source through the specimen and to the objective lens. The Darkfield microscope. In a darkfield microscopic image, light objects are visible against a dark background. Light is reflected off of the specimen and enters the objective lens, following the process seen on this slide to the bottom left. The image that you would see in darkfield microscopy is seen at the bottom right. Phase contrast microscopy is illustrated here. The process is broken down in the top right-hand corner of this slide. Phase contrast microscopy is designed to accentuate the diffraction of light that passes through a specimen to provide higher contrast and higher resolution than simple bright field microscopy. If you examine the picture at the bottom of this slide, you should be able to see a notable difference between bright field microscopy, that which is produced using a simple light microscope, and phase contrast. Fluorescent microscopy uses ultraviolet light. Fluorescence substances absorb UV light and emit visible light. Cells may be stained with fluorescent dyes called fluorochromes. An image of what fluorescent microscopy would appear like through a microscope is shown here. Confocal microscopy is a lot like a CT scan for bacteria. In confocal microscopy, the microscope produces images as slices. The electron microscope. The electron microscope uses electrons instead of light. Electrons travel at a shorter wavelength than visible light and therefore produces greater resolution. Electron microscopes can be transmission or scanning. The transmission electron microscope, or TEM, utilizes ultra-thin sections of specimens. Light, or in this case electrons, will pass through the specimen, then an electromagnetic lens, and then to a screener film. Specimens may be stained with heavy metal salts, and a magnification of up to 100,000 times with a resolution of 2.5 nanometers can be achieved. The scanning electron microscope, or SEM, is illustrated on this slide. The process of scanning electron microscope and an image produced by one can be seen to the right. Scanning electron microscopes use an electron gun to produce a beam of electrons that scans the surface of a whole specimen. Secondary electrons emitted from the specimen are then used to produce an image. A magnification of up to 10,000 times with a resolving power of 20 nanometers can be achieved. This slide simply compares the transmission to scanning electron microscopes. Transmission electron microscopes have greater magnification but only produce two-dimensional images, while the scanning electron microscope suffers in overall magnification. It can produce a three-dimensional image. Some other types of microscopy include two-photon microscopy, scanning tunneling, and atomic force microscopy, each serving their own purpose in achieving a magnification that in some cases can make it possible for us to see almost all the way to the point of an atom. Preparing specimens for light microscopy. Microbiologists will utilize one of two scenarios when preparing specimens for light microscopy. In one case, wet mounts may be used. A wet mount involves taking a specimen and placing it on a slide in a drop of liquid. These live or unstained cells often have little contrast with the surrounding medium, which makes them difficult to see. However, by using a wet mount, we can get an idea of their behavior in their natural environment. Fixation attaches the specimen to the slide and kills the specimen. Killing the specimen provides us with an opportunity for long-term storage and the ability to stain the specimen. Staining and fixing is illustrated here. When staining and fixing a specimen, the specimen will be smeared to the slide in a thin film of a solution. That slide will then either be heated using a Bunsen burner or fixed using chemicals. Staining. Staining is the coloring of the microbe with a dye that emphasizes certain structures. Some stains will stain the organisms themselves, while others simply stain the background the organism is in. Stains are salts composed of a positive and negative ion, one of which is colored and referred to as the chromophore. In basic dyes, the color is the positive ion. In acidic dyes, the color is the negative ion. Bacteria, in their natural state, tend to carry a slightly negative charge, and therefore will attract positively charged basic dyes such as crystal violet, methylene blue, malachite green, and safranin. Acidic dyes are repelled by the bacteria and subsequently color the background, but not the cells themselves. Simple stains. Simple stains involve the utilization of a single stain, and as that stain is used, all the cells in a smear or on a slide are stained. Aqueous or alcohol solutions of a single basic dye are used in this process. The stain is applied to this fixed smear and then washed off a short time later. The use of simple stains ultimately involves processes meant to determine the shape of cells, the arrangement of cells, and to estimate the size of cells. Differential stains. Differential stains react differently with different kinds of bacteria and can be used to distinguish amongst them. Sometimes a special treatment or chemical is used to set the stain. We refer to that chemical as a mordant. The Gram stain is the most frequently used differential stain. Gram-positive and Gram-negative are the two major categories of bacteria that we are able to sort through by using a Gram stain. An acid-fast stain is another type of differential stain. Gram staining was developed in 1884 by Hans Christian Gram, and it differentiates bacteria into two large groups, the Gram-positive and Gram-negative, based on the microorganism's cell wall reaction to the staining process. All the cells are stained with crystal violet, destained with alcohol. Gram-positive cells will stay purple, while Gram-negative cells will lose their color. The decolorization or destaining process can wash the crystal violet off of a Gram-negative cell, but not off of a Gram-positive cell. The cells are then counterstained with safranin. Safranin, being pink, will be hidden by the copious amounts of crystal violet that remain after the decolorization step in a Gram-positive cell. Gram-negative cells, however, have lost that crystal violet and subsequently will only absorb this counterstain in the final step of Gram staining. When looking at Gram-positive cells under a microscope, you should expect to see purple cells. While looking at Gram-negative, you should expect to see pink cells. This is an illustration of Gram-positive and Gram-negative cells. Acid-fast staining is another differential stain. It is used for Mycobacterium nocardia. These organisms have a waxy cell wall, which is difficult to destain. Cells are stained with carbofuchsin, which is pink, and decolorized with acid alcohol. Only acid-fast cells stay pink. The rest are counterstained blue. This table illustrates the steps and the appearance of different types of acid-fast cells. Negative staining. Negative staining is useful for capsules because it stains the background and not the organism itself. Illustrated here are two slides that are negatively stained. The endospore stain. Endospores are microbial lifeboats that contain genetic material inside of a cell, oftentimes produced as organisms are exposed to environmental conditions that might result in their demise. Endospores are very resistant structures. They are produced copiously by the genera Bacillus and Clostridium. Heat is required to drive an endospore stain into an endospore. In this picture, you can see the green stained endospores inside the pink or red bacterial cells. And finally, we have the flagellar stain. The flagellar stain requires a mordant and is designed to stain the thin hair-like projections that some bacteria use for locomotive purposes, referred to as the flagellum. This is probably the most challenging stain in a microbial lab environment. And this concludes our coverage of Chapter 2.